I'm very happy to be here today to speak to nutritionists. Disclosures. Largely, I'm interested in getting valid claims out of data, so that's my main intent. I have worked in industry, worked with Eli Lilly and PepsiCo. Strategies to improve the reliability of observational studies. I gave a short talk at the NIH one time, and I was a little nervous about who I was talking to. So I said, how many people here worry about the reliability of observational studies? Now, these are the people that fund a great deal of this research. I'd hardly gotten the words out of my mouth, and every hand was up. So I felt the audience was at least a little sympathetic to what I was going to be saying. I could tell you about the meaning of life, but we really don't have enough time today to do that. What is real? I'll leave that to the physicist. What is reproducible? That's what statisticians are largely all about. The claims you make, are those reproducible? And there have been some interesting books on fooled by randomness. Who's the easiest person to fool? Yourself. So you need to follow specific procedures so that you don't trick yourself, and then I'll talk a little bit more about that. It's interesting, the players have sort of changed over the last 5 or 10 years. The workers, those are the scientists. We'll be talking about epidemiologists a lot today. The communicators, I'd never thought too much about this. Science writers are a dying breed. They've been gone away. In their place, PR people will write press releases, and reporters will reword those press releases, and that's what we largely see in the popular media. Consumers, we're all consumers. We want reliable results when we're reading things. As a scientist, you're producing this and that, and you can only produce so much. But as a consumer, you see lots of things. I'll talk a lot about management. Funding agencies and journal editors. Who controls the scientific process? These are the managers of that process. Deming went to Japan after the Second World War and revolutionized industrial production. He was the most visionary innovator on quality control that we've had so far. His comment is, the worker is not the problem. The problem is at the top. The problem is management. Workers work to a procedure, they respond to incentives, and if the incentives are not good, they will respond to them, and it's up to management to change the incentive system. So I'll speak today a lot about management. Problems with observational studies. I just wrote this down very quickly. The first one really I don't think is known and appreciated as much as it should be. When you get raw data, you have to sort of clean it up and shape it up and get it ready to go into statistical analysis. That is a very laborious task, and there's lots of flexibility about how you do that. It turns out that that part of the process can dramatically change the answers that are coming out. The rest you all know about. Often in nutritional and epidemiology studies, there's no written analysis protocol. Tens to hundreds to thousands of questions can be at issue. Multiple modeling. You've got covariates. You can bring them into and out of the analysis. Uncorrected bias. People love to talk about uncorrected bias because mostly you can't do anything about it. They really don't like to talk about multiple testing and multiple modeling. Self-serving paper writing. Is the purpose of the research to get a paper out, or is the purpose to make a reliable claim? Those are two different things. Self-serving press releases. How many scientists actually go over the press release and say the press release is a fair representation of what is in the paper? How many reporters or science writers actually read the scientific paper, or do they just work from the press release? The other one is purely psychological. People get involved in their work, and they should. But at some point, they believe their work so much they become an advocate rather than more dispassionate. Data staging I've already talked about. Multiple testing I'll spend most of the talking about. Multiple modeling and bias are difficult topics, and any or all of these things can be used to make a study positive. I asked David Madigan, a statistician at Columbia, a question: why do you think data staging is a big issue? And he said, because it can be done in a myriad of ways, it is rarely documented, and is usually not reproducible. So if you ask the same group to stage the data set again, you'll get a different data set. Okay, I'm a statistician. We'll do a little bit of statistics here. The basic premise of a t-test is you have two groups that you want to compare, here and here. You presume that they came from some mythical superpopulation, and you compare the mean values for these two groups with a t-test. Simple enough. However, if you do enough t-tests, you're guaranteed to have a statistically significant result. So for all of you that need statistically significant results, here's the slide that helps you out. Don't do one comparison. Don't do two. Move all the way up to about 60 comparison, and there's a 95% chance you will have at least one statistically significant result. You can write your paper, you can get your thesis, you're on your way. I've run this experiment with lots of audiences. I give the people 10-sided dice. If you play Dungeons and Dragons, you'll have seen these things. Red, white, and blue. We're all very patriotic. We're here in the home of patriotism, Boston. If you roll these dice and write down the digits that show up on them, you can get a p-value. So let's run an experiment. Okay, we've run an experiment. Every time we do this, we have different side effects, and we have different types of people, and I've blocked in red 3 p-values that are less than 0.05. Heck, this is a lot easier than running a real experiment, but it is a mimic of a real experiment. If you ask 61 questions, there's a good chance you will have at least one statistically significant result. Here, you can actually point to two things in the same thing: stroke and young and young males and old females. Now, the sort of the statistical deconvolution of what I just did. P-values are supposed to be uniformly distributed between 0 and 1. So if you rank order the p-values from smallest to largest and plot them against the integers, if that turns out to be a 45-degree line, then the p-values you're looking at are from a uniform distribution. So this is really just sort of a joke. I mean, who would do something like this? Only a statistician would do something like this. But in fact, if you look at the literature, quite often there are a large number of questions at issue, and if any of those questions come out statistically significant, a paper can result. Well, you'd say, well, who would do a thing like this? You are what your mother eats. Wow. Gosh, mother's coming out of the grave to talk to me. Evidence for maternal preconception diet influencing fetal sex in humans. It's a joke. I mean, why would eating cereal produce more boy babies than girl babies? This appeared in the Royal Society B. This is the British equivalent of PNAS. This is a prestigious journal. Well, 3 friends and I, we looked at it and we said, you know, this is a bad joke. It's a joke, but it's a bad joke. So we asked for the data set, and after 6 months of cajoling by the editors, the data set was turned over to us after we signed a 6-page legal document. And this is after the authors of the paper had promised to give the data set to any reasonable person. Okay, well, now you can laugh. Stan Young is not a reasonable person. You should give me the data sets, but it took a while. So we wrote a response: gender-induced selection, most likely a multiple testing false positive. Here's the p-value plot. There were 262 questions that were asked, and the authors focused on one of these little p-values down here. But if you look at the whole distribution, it's uniform. There was no evidence here. And the stream of emails back and forth to the editors was interesting, but I won't go into it. Science in a Crisis, 2011-2012. We wrote a paper, Deming Data and Observational Studies, and a lot of this talk comes from that paper. The second paper here, though, is much more disquieting. Raise standards for preclinical cancer research. Okay, so this is experimental biology. The scientist has complete control over everything that is going on. Observational studies, I think most people agree, have their problems. Well, here's the observational studies, and I'll walk you through this. We found 12 studies where an observational claim was tested in a randomized clinical trial. So we moved from observational data to presumably the gold standard of science, randomized clinical trials. This gives the number of claims that were at issue in the trial. This gives the number of claims that were substantiated. See a lot of zeros. None of the claims were substantiated. Every single one of these 12 studies had rather massive observational data saying that the claim was reasonable. This number right here is pretty interesting. Where is it? Yeah. 3. Hormone replacement therapy. Initial observational studies said that hormone replacement therapy is a good thing. This study came up with 3 problems, side effects that were untoward. But if you look at this number down here, 5 out of 52 is sort of close to 5%. It's more than that. But even the claims in the negative thing could be random when looked at in the body of the evidence. So the initial studies on hormone replacement therapy were probably wrong in the sense they said it was beneficial. And right now, I don't even think we have evidence to say that hormone replacement therapy is detrimental. The Nature study, there were 53 claims that appeared in best journals, Science, Nature, Cell, so forth. Of the 53 claims, 47 of them could not be replicated. All right, this is in experimental biology where the scientists can control everything. They can run the experiment again, so forth and so on. So if scientists, where they have control over everything, reach the wrong conclusion about 90% of the time, then we as consumers need to be particularly vigilant. Okay, I'm going to pick on funding agencies for a while. Funding agencies should fund the generation of observational data sets and the analysis of those data sets separately. So you fund one group to build the data, and then you make the data public, and then anyone or any group can analyze the data. If you tie the two together, there is the interest of the investigator to keep the data private as their own little gold mine. So funding agencies should fund data generation and analysis separately. There's very little funding of replicate studies. Given that the error rate or the lack of reproducibility is so high, I think they should start funding some replication studies. And then funding agencies should require that when a paper is published, the data used in that paper be public. What can journal editors do? The biggest thing that they can do is, we'll go back to Deming. quality by inspection. 50 years ago, we would make widgets, we would inspect them, and if the widget was a good widget, we'd sell it. If it was a bad widget, we'd throw it in the scrap heap. This was called quality by inspection. It was a particularly ugly way a very costly way to make things. Deming came in and said you need to look at every step of the production process and make every step high quality so that the only thing coming off the assembly line are high quality widgets. And then you sell them all. Journal editors, to a large extent, look for a statistically significant result, a p-value less than 0.05. It's quality by inspection. If you don't pass that, your paper doesn't get accepted. If you do pass that, you have a chance. Journal editors should be looking at the whole quality of process that's going on and accept papers if the work is good, irrespective of whether it has a p-value less than 0.05. And I will say that many workers have figured out how to game the system. Multiple testing is one way to do that. Management needs to redesign the system to build quality into the product. There's more that could be said there. Consumer, what can you do? The answer is really not very much. There's a stream of stuff coming at you that you read. There's not much you can do. I would say be skeptical of observational studies. Read the actual paper if it's of interest. You can do the following thing. You can attempt to count the number of claims that could be made from a paper and the data of this sort. If the number of claims is very large and you see not too many small p-values, then you should be pretty skeptical. You can ask for the data set. My experience in asking for the data set is you usually find out they just won't give it to you. Okay? And that I think is a thing to worry about. Write letters to the editor, voodoo statistics and trust me science. So if they ask 100 questions, won't give you the data set, and report a p-value of 0.03 as being real, that's voodoo statistics, and they won't give you the data set, that's trust me science. The letter you write is really not to address the worker. The letter you write is to alert the editor that there's a potential problem, because editors can fix this, workers, according to Deming, will not. You can write to the funding agency, and you can talk to your congressman, which I've done on occasion. Okay, we're going to do a new p-value plot. Now, over on the left, we have a p-value plot. There's a lot of repetition here, but this is like a sort of a 50% line here. These are the little guys, the little p-values, and we want to know if those are real. Well, what we've done is we've taken the p-values and we take the negative log of the p-value. Okay, negative log, remember back in high school. So you take the negative log, these numbers now show up on the upper right-hand side. If there's no, and we plot those against the expected values. So a little bit of mathematical statistics here. You work out the expectations of the negative logs of the p-values, and you put those here. If you're on a 45-degree line, nothing is happening. If you fall off the line, you can see these guys that are sort of obscured down here in the corner. Here, the evidence is that one of these claims is likely to be, there's a real effect. These two are problematic, but most of the claims are not effect. These were side effects in a clinical trial. Here's an interesting one I just picked up the other day. 2 cancer types, prostate cancer, aggressive and non-aggressive, 48 pesticides. They were looking at workers exposed to these chemicals. There are a total of 96 questions. The paper, wonderfully for me, gave all the p-values. So here they are in a p-value plot, and that looks pretty much like a 45-degree line, but down here the small guys are the ones that we're interested in. The authors actually made 3 claims. If you replot this with the new p-value plot, you find that these two small p-values here are not very far off the expected line. Those are probably false positives. This one is off the line, so of the three claims that they made, it appears that only one of those claims is statistically justified. Conclusions. Most science claims do not replicate. Now, there are a few hundred people here. Your claims all replicate, I'm sure. Okay? But it's these other guys that you've got to watch out for. It pays to pay attention to Deming. When things go wrong, you really shouldn't be blaming the worker. People here are probably managers, and so it's a little embarrassing. Although your claims are always right, you're probably managers, and you need to get involved in the situation. Funding agencies and journal editors have largely been absent without leave. Requiring the data to be placed in a repository allows the normal science functioning to operate. If people are allowed to keep the data private, the speed with which we can progress science is greatly attenuated. And I think, yeah. I'll make one comment. Office of Science, Technology, and something, OSTP, February 22nd, is effectively going to mandate that any federally funded research, the data used in that research or in a publication, become public. So people should take a look at that and figure out how they're going to react to it. And I think that's it for me. So thank you very much.